discussing? We just discussed about the important regulatory protein, and then we discussed the role of P53 protein. If there is a DNA damage, then how the cell will check, or how there will be checkpoints will ensure that there must be proper DNA replication. Otherwise, they will arrest the cell cycle. So now I'm going to so am I audible?
Yes, ma'am. Yes.
Okay. Sorry for the inconvenience. So we have to discuss about basically the S phase. Today we will discuss about the S phase. After that, we will move further to the different type of cell division that we will discuss. You guys told me that we have to discuss about the mitosis and meiosis. So first of all, I will sharing my screen. I will share my screen. So let me know screen is visible to all.
Yes, ma'am. Yes.
Okay. So S phase we are going to discuss. S phase is why the name is S phase. S for synthesis phase.
Synthesis.
Yes, synthesis phase or replication phase. So what will happen in the S phase? Replication takes place in S phase, and how it will happen we are going to discuss. So firstly, if this is a DNA and this is a conserved region of DNA, which particular sequence will be there? This is the sequence that is origin sequence. In eukaryote, we use one term for this particular sequence. Where from where the replication starts in eukaryotes? Which from?
Origin of replication.
Are you sure? Origin of replication or ARS, autonomous replicating sequence, present in the eukaryote. So what will happen in the S phase? Firstly, there will be helicase loading. Helicase will load here, and helicase loading will take place in the G1 phase. After that, the cell will come in the S phase. In S phase, firstly, there will be binding of or loading of origin recognition complex that made up of 6 subunit. Origin recognition complex will load here. After loading of origin recognition complex, there will be loading of MCM2, MCM227. Here there will be loading of MCM227. And then there will be loading of CDT1, CDC6, CDT1, 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 and then CDC6 at this particular site. So after these different loading, this whole complex is known as pre-replication initiation complex. Pre-replication initiation complex. So helicase, as we just discussed that helicase will load in the G1 phase of the cell cycle, but in the S phase, there will be activation of helicase. Helicase will get activated in the S phase of the cell cycle. Now, what will happen after loading of MCM, CDT1, CDC6? There will be activation of helicase, and how helicase activation will take place with the help of CDK2 and with cycline, CDK2 and cycline A. The role of this complex to activate the helicase. Am I clear? You all are getting?
Yes, ma'am.
Okay. So this CDK2 and cycline A, their role is to add phosphate group on the accessory protein. What are the accessory protein? This MCM2, CDT1, CDC6, they are the accessory protein. So the CDK2 and cycline A will come and it will add phosphate. How? Why this particular CDK2 and cycline A is doing so? Because there will be, sorry, there will be, there must be opening of this ARS sequence. So CDK2, cycline A and DDK kinase. There is one kinase, that is DDK kinase, will come and it will add phosphate group on the MCM2. This will, DDK kinase will add the phosphate group on MCM2, and the CDK2 and cycline A, they will add the phosphate on the CDT1 and CDC6. So what will happen if there will be attachment of the phosphate group onto this? They will, particularly they will be removed from this site and there will be formation of replication bubble. Replication bubble, you all are aware of this. Replication bubble.
Yes.
So you have seen the leading and lagging phase in this replication bubble. Now, so a replication bubble is just opened. So there will be in S phase, there will be replication. Now we have completed the S phase. Now we will move on to we will jump to the G2 phase. I hope there is no doubt to anyone. If there is any doubt, please let me know.
Yes, ma'am.
Yes.
Ma'am, how is that replication bubble is forming? I mean, is it
Replication
because of the removal of
melting of, basically there is helicase. As you know, what is the role of helicase?
to unwind the DNA?
So it will unwind the DNA from that particular location by just breaking the hydrogen bond. As you know that the two strand of the DNA, this two strand or helix of the DNA, they are attached with the, or they are just in the form of this particular helix because of the hydrogen bond that is present between the two helix. So the role of this helicase is to break this hydrogen bond, and this process is known as melting of DNA. Okay, and then there will be just formation of replication bubble. Am I clear? This whole complex, this whole process is very complicated, but I have given you a brief introduction, brief explanation of this particular thing. Now G2 phase is known as the preparation of mitosis. Mitosis preparation will take place, microtubule biosynthesis will take place. Microtubule biosynthesis, you all know what are microtubules? Microtubules?
Yes, ma'am.
Yeah.
Aren't they used during the metaphase for like separation of like alignment of the chromosomes in the plane?
Any other answer for this? Okay, now tell me, microtubules, they are the part of, no one.
Spindle formation assembly.
Okay, spindle formation assembly. They are the part of cytoskeleton. Yes, cytoskeleton. And they are made up of tubulin protein. Tubulin protein is there, and These microtubule polymerization, depolymerization, you guys have heard. So that is basically microtubule biosynthesis takes place in the G2 phase. And MPF, MPF we had discussed in the previous classes. MPF, mitotic promoting factor, mitosis promoting factor. And this mitosis promoting factor, we have seen it is made up of cyclin B and CDK1. That mitosis promoting factor formed in the G2 phase of the formed before the G2 phase of the cell cycle, but at the end of the G2 phase of the cell cycle, it gets activated. This mitosis promoting factor or maturation promoting factor, we just discussed that CDK2 and cyclin 1, they are forming one complex MPF, and with inhibitory kinase, in the presence of inhibitory kinase, that was V1 kinase, that add the phosphate on the threonine residue, 14 threonine 14 residue and tyrosine 15 residue. Because V1 kinase is inhibitory kinase that just attach the phosphate at the inhibitory site. Threonine and tyrosine, they are just inhibitory phosphate. So that mitosis promoting factor gets inactive, but in the presence of activatory kinase like CAK, in that we just discussed the CDK7 and cyclin H, they form the cyclin activating kinase, sorry, CDK activating kinase, and this will just add the phosphate on the threonine 161 position. So what will happen? Now again the mitosis promoting factor is inactive. Then in that particular scenario, your CDC25A will come, that is activatory phosphatase, and it will remove the phosphate from the threonine 14 and tyrosine 15. Then MPF get active. And what will happen if MPF get active? Now, see, this whole process is clear. This is a quick revision of the previous class, what we just discussed about the V1 kinase. As I told again and again, there will be repetition of the same thing. So I hope it is clear. Clear
Yes,
now?
ma'am.
Okay. So this is the CDK. Now this one is the cycline. Okay. So at threonine, at threonine 161 position, there will be phosphate group. Now this CDK and cyclin 1, that is forming the MPF. Now this MPF is active. This positive I am writing for just active protein. Now, what will happen? This MPF will there is one Aurora kinase. Have you heard about Aurora kinase? Aurora kinase.
No, ma'am. No, ma'am.
No. Okay, leave that. See, this MPF will act as a kinase here. And what is the role of this particular MPF? It will do the this will act as a kinase. And it will do the phosphorylation of condensin. Condensin, have you heard? Condensin. What is the role of condensin? Anyone? Condensation of chromosome, you all have heard. Okay, non-histone, yes.
Yes, ma'am.
So condensation of chromosome takes place in the presence of this condensin protein. Then cohesin,
Yes, ma'am.
phosphorylation by MPF. What is this cohesin? Cohesin, what is the role of this cohesin? If this is one, see one, this is one chromatid, this is another chromatid. So these two
It holds
chromatid,
two sister chromatids.
hmm, yes,
It holds the sister chromatids together.
yes, cohesin. That is the role of cohesin. condensin within the same particular chromosome, and cohesin in between two different chromosomes. So cohesin get phosphorylated, condensin get phosphorylated, and with this there will be chromatin condensation. Chromatin condensation. Now, the role of this is one role. Another role that it will do the phosphorylation of centrosome. Centrosome, centrosome. Yes, anyone? Centrosome. It will do the phosphorylation of kinetochore. Kinetochore, you all have heard. Centrosome, then microtubules, associated protein. So centrosome, have you heard about centrioles?
Yes.
Yes.
So that is what centrosome, na? So it will do the phosphorylation of centrosome, kinetochore. The one protein is present beside the centromere, that is kinetochore, on which uh, protein the spindle fiber get attached. That is kinetochore. So kinetochore get phosphorylated, centrosome get phosphorylated. Then another role is nuclear lamins get phosphorylated. Then nuclear pore complex get phosphorylated. Inner nuclear membrane get phosphorylated, so that there must be nuclear membrane breakdown. Then Golgi matrix protein get phosphorylated, so that there must be fragmentation of Golgi. Then H1 protein get phosphorylated. So this is the role of this MPF. See, it will do, as I told, it is a kinase. So it will do the phosphorylation of different protein present in the cell. Am I clear to all? Clear? Now we are going to start the prophase. We are going to start the cell division. For that, we are going to start with the prophase. So what do you understand by prophase? Prophase is what?
First phase wherein the organelles start to disappear. Nuclear membrane starts to disappear.
Yes. Any other answer for this? Yes. Any other answer? No. Okay. So first phase is chromosomes in condensed state. See, in prophase, nuclear membrane fragmentation will take place, that we have seen the MPF activity. In particularly in MPF, you will find out that MPF, there will be activity of, basically not in MPF, sorry. In prophase, there will be activity of MPF. You will see that MPF get activated and it will do the nuclear membrane fragmentation, genetic material condensation, microtubule formation initiate from the centrosome in prophase stage. Then what will happen in metaphase? What will happen in the metaphase?
Arrangement of chromosomes in
Arrange.
a central plate.
At the equator plate.
Yes, equatorial plate. So maximum chromosome condense form genetic material, and this chromosome comes at the middle. If this is a cell, so they just align at the equator. All these chromosomes will align here. That is the equator. And then anaphase, what will happen in the anaphase?
Separation of the chromatids.
Separation of the
Chromatid-
chromosome, simply just, yes, separation of chromosome. Now we are going to discuss about my cell division particularly. So firstly, karyokinesis. Have you heard about karyokinesis?
Yes, ma'am.
What is karyokinesis?
Nuclear
Division of nucleus.
Nucleus. Mitosis takes place in the somatic or stem cell. Meiosis takes place in the germ cell. So in mitosis, there are different phases like prophase, metaphase, anaphase, and telophase. What will happen in early anaphase? This MTF activation phosphorylate the cohesin, condensin, phosphorylation of Golgi matrix complex, nuclear membrane, and then what will happen after that? Metaphase, all the chromosome will align at the equator. Sorry for my bad diagram presentation. Okay. And then in anaphase, what will happen? In anaphase, you will see in early anaphase, we are talking about the mitosis. Remember that. So in early anaphase, there is one protein that is APC plus CDC20. Have you heard?
Yes, ma'am.
What are their roles? Yes, what are their roles? Anyone?
Ma'am, it helps in promoting the occurrence of anaphase.
How? See, this is anaphase promoting complex or anaphase protein complex. In early anaphase, this protein complex APC plus CDC20 get activated. This APC and CDC20 gets activated. Now, how it get activated? I'm not going detail of that particular thing, but it will form active anaphase promoting complex. See, as the name suggests, it is a anaphase promoting complex. So, have you heard about securine and saparase? Securine and saparase. This securine, this anaphase promoting complex will do the phosphorylation of securine. Am I audible?
Yes, ma'am.
Okay. So what is the role of securine here? Anyone? See this saparase, saparase is, what is the role of saparase here? Saparase, sister chromatid we have seen. We have seen the sister chromatid. So both sister chromatid, they are attached with which particular protein is playing a very vital role here? Cohesin or condensin?
Cohesin.
Cohesin. So the role of this saparase, it is acting as an enzyme here, and this securine is inhibiting the saparase. So saparase is actually degrading the cohesin. It will degrade the cohesin, but with securine, it is inactive. So that's why anaphase promoting complex do the degradation of securine by ubiquitination. We just discussed the ubiquitination. It will attach the ubiquitin protein and it will just take away the securine to the 26S proteasomal complex. Now separase get active in the absence of securine, and separase it will degrade the cohesin present between the two sister chromatid. And then what will happen? Two sister chromatid, they will be apart from each other. Am I
Yes,
clear?
ma'am.
Yes, ma'am.
Okay. So, like this, there will be separation of the sister chromatid in anaphase in the presence of separase. Clear to all? Now, telophase.
Yes, ma'am.
Telophase is what? In telophase, what will happen? Telophase.
Ma'am,
Yes.
during the early anaphase, this takes place. What happens during the late anaphase?
There will be MPF degradation. MPF will be degraded, and how MPF get degraded, because as you know, MPF is made up of CDK1 and cyclin B. There will be degradation of cyclin B. As I told earlier also, that cyclin level will fluctuate within the cell during different event of the cell cycle, but CDK will be present as it is, that CDK1 will present in the cell but in inactive form. Okay.
Okay, ma'am.
So what will happen in the telophase? Yes, telophase. No one. Telophase.
Separation of the daughter cells.
Separate. Yeah.
Separation, and how this separation will take place, you will see at the middle, there will be formation of microfilament. That basically actin filament will just form here between the cell, and it will form two different cells by cytokinesis. Am I clear?
Yes, ma'am.
Yes.
Okay. So with this, we have just discussed the mitosis. We have to discuss about the meiosis. So what do you want? You want the meiosis discussion today or tomorrow? Not tomorrow, on Monday.
Monday.
Monday. Okay, please do revision of the classes that we just discussed and enjoy your weekend. We will meet on Monday. No classes on
Thank you,
Saturday.
ma'am.
Bye bye everyone. Good night.